a audience that's visible, so I will not bite more into your time. Hello, everybody. Welcome this morning. It's one of these exceptions. Normally, we do this seminar in the afternoon because that's better for us Europeans here. But we want to facilitate a broader participation base. So we do this in the morning so that our speaker Sam can join us without having to stay up until 2 a.m. So this seminar is going to be interesting. It's going to be about an interesting group of creatures, ants, which is one of the groups that Sam has been studying. Sam is currently postdoctoral fellow at the Institute of Molecular Bioscience at the University of Queensland, Brisbane in Australia. And he has been studying venoms in a whole range of organisms, including plants, but also several animals, in particular cone snails and ants. And I think he may give a few more words about this when he starts talking. And I think, what Sam is particularly interested in is pain, especially how venom components can elicit pain in organisms, and what you can learn about pain mechanisms, and perhaps even exploit this knowledge to use, for instance, venom components to come up with new treatments for pain and other afflictions in humans. So today, Sam is going to talk about ants. If you haven't read some of his works, he's been quite prolific. And a recent paper I think I want to highlight was published not that long ago in BMC Biology, together with Ivan Undheim and a range of other authors. And it was about, and I cannot pronounce this species of ant, an Australian species of ant. And it's a very interesting ant where the workers have distinctly different venom profiles, one from another, but then together, this creates this, what seems to be a very adaptive colony-wide venom phenotype that is very useful for various purposes, defense and predation. So this paper in BMC Biology, Sam is the first author. I urge you to look this up and read it. It's a really fantastic, and I think conceptually really novel way of looking at venom. So congrats about that paper, Sam. I've talked enough. I'm just happy that you can join us here this morning. Look forward to your talk, and the digital platform is yours.
Thank you. I will now share my screen. Where are we? All right. So thanks, Ronald, for the introduction, and thanks everyone for joining. I can see a few familiar names there. And hopefully I can give you a good talk for the next half hour or so. So as Ronald said, I'll be talking about the defensive chemistry of ants, and one sort of more recent story that's still mostly unpublished, and I hope you find it interesting. So before I jump into that, I'll just give you a bit of background, because I always appreciate having sort of knowing a little bit about the people talking. So actually, I'm originally from New Zealand, way down in the corner of the world. I completed my BSc honours in 2008 at the University of Auckland. And then I worked as a research technician in molecular neuroscience. And during this time, I sort of discovered toxinology. I was working on sort of mapping these pathways in neurons and using these natural products as a sort of a reductionist thing to block off specific pathways to figure out what was how things worked. And I think the original one I came across was from a cone snail. And I thought, this is the coolest thing ever. I was thinking about if I wanted to do a PhD, and I thought, yeah, I could spend 4 years studying these toxins from cone snails. So I wrote up a little proposal, and I ended up going over to MIPS, or Monash Institute of Pharmaceutical Science in Australia, to work with Ray Norton. And I worked on a cone snail, the one pictured there, Conus victoria. And my goal right early on was I wanted to look right from the beginning to understand what was in the venom of the snail, so venomics. And back then, this was really sort of early venomic stuff. And then make some of the toxins, functionally characterize them, and then structurally characterize them. And I remember Ray told me afterwards that he thought I was a bit ambitious to start with, and he was amazed that actually by the end of my PhD I was able to do all of that. And then once I'd finished that, I was able to go over to the University of Utah to to join the Oliveira lab and as a postdoc for a couple of years. And as you can imagine, I was able to work on, instead of one species of cone snail, a whole range of species, which was great fun. And while I was there, I sort of got back into the neuroscience side of things. They were using toxins from these snails to understand, they were actually using sensory neurons to understand what these toxins were targeting. And then after that, I moved back to Australia and I needed to develop a program of my own. The cone snail field is pretty crowded, so I wanted something a little different, but still using the skills that I developed. So then I returned back to neuroscience, specifically the sensory nervous system. And I wanted to use toxins as tools to sort of understand how our sensory nerves function. And I've coined the name sting science.
Sam, can I briefly interject for a moment? It is you have you have presenter view on, so we can also see next to your slides the upcoming slide in a smaller format.
Okay, let me see if I can swap that.
I think maybe in the chat somebody suggested, can you put in full screen mode?
How's that?
That's perfect. Now we don't see what's coming, and then the surprise is maximized.
Yeah. All right. So yeah, so about my research program, I can really boil it down to sort of one question, and that is, why do stings or bites from venomous things hurt? And that's sort of, I can split that up into three things. So that is understanding the chemistry behind stings. What's the pain-causing chemistry? Understand the pharmacology. So what are those pain-causing toxins doing to our bodies for us to sense that pain? And then, of course, the big why of why do these things hurt is actually, why have they evolved to do this? And this is enough for me. This is what excites me. But as I said, you always need sort of an underlying translational thing. If anything, it's to get funding. So what I think this can be helpful with is, we know that venom toxins are really useful tools for studying cardiovascular system. They've really helped with understanding the neuromuscular junction. But I think because they've evolved to cause pain, why not use them to understand the sensory nervous system as well? That's what they've evolved to target. And what I mean by that is, different toxins might be targeting different components of our sensory nerve endings. So for example, different ion channels or receptors involved in pain signaling. We might be able to use them to identify new pain targets. So a new ion channel that we didn't know was involved in pain. And actually, we've actually just published that exact thing about a week ago. So Irina Veda discovered a new subunit for a sodium channel, voltage-gated sodium channel. So from the stinging tree, a stinging tree toxin. And ultimately, the goal is perhaps, I envision that perhaps we'll be able to, with identifying these new pain targets, we might be able to come up with new treatment strategies for pain, of course. So there's lots of things that sting and lots of things that hurt when they sting. So the question was, where do I begin? And this is a picture from Vanessa Schendel's review. And you can see this illustrates all the different things that sting defensively. And really, when I moved to Australia, I started with ants for reasons I won't explain. And that was with Ivan, Glenn King, and later joined by Axel Touchard, who's really sort of the expert on ant venoms. I've also worked on assassin bug venoms with Andy Walker, all around this question of what are the components or the toxins that sting. Stinging trees, as Ronald mentioned, with Tom Jurek and Irina Veda. Spitting cobras with Nick Casewell, caterpillars, again with Andy Walker, and other hymenopterans with Justin Schmidt and also with Axel Touchard. But today I'm just going to cover the ants. So this is where we started. Ivan and I wanted to look at the venom of this particular ant. We didn't really know how. So we sort of came up with this way of collecting the venom. And as you can see, it's pretty simple. We just hold the ant and they're quite happy to sting this parafilm. And, this is pretty commonplace stuff now. It's essentially what you do with milking some snakes. And you can see little droplets of venom collecting on that parafilm. So we're getting this nice pure venom. We can just rinse that off with a bit of water. And then once that's collected, then we can analyze it by LCMS. And I'm not going to go into the details of how we do the venomics. I think that's everyone sort of probably has a pretty good understanding of that. But basically, my goal is to identify what these peaks represent in the venom, and also understand where those sit within the transcriptome. So what we found was that obviously the venom is not formic acid. Probably shouldn't come as a surprise to most of this audience, but you'd be surprised, not everyone. But in fact, that it's peptidic, and it's also relatively simple, at least compared to, say, some of the cone snails I've been working with. And what you've got here is just a snapshot of what's in that venom, and you can see they're all peptides. And actually, all of these ones are sort of, if you stare closely, there's some similarities in their structure. And we looked at the precursors of these, and it looks as though they may be homologous, so they're derived from a common ancestor. And we called them, sort of hypothesized that these might be a gene superfamily, and we called this the toxins. And these toxins in general are peptide. They're all peptides in general. These peptides are cysteine-free or have very few cysteines. They're often quite hydrophobic, but also have cationic residues. And that allows them to form these amphipathic alpha helices. So you see here the hydrophobic residues on one side of the helix and the cationic residues of the other. And this makes them membrane active. And a really classic example of this is melittin, the main component from honeybee venom. And what this allows them to do is form pores and membranes. If this is a neuron or another excitable cell, it will depolarize the neuron. This will cause an action potential, and then that causes pain. So that's what we found was going on with at least the major component in the bull ant venom. So in some ways, this was a little bit disappointing. It didn't tell us anything new about pain or anything new about any of the channels involved in pain. We know there's membranes in neurons. But there are ants that definitely have very special stings. So some of you might have heard of the Schmidt's sting pain index. And this is just a really nice graphic sort of visually illustrating it. What you can see right here in front and center, shown big because it has the most painful sting, is an ant. So this one is Paraponera clavata, or the bullet ant. And there are other ants here too that have really interesting stings. And Justin described this sting as pure, intense, brilliant pain, like walking over flaming charcoal with a 3-inch nail embedded in your heel. So there's something special about it. It sounds like it's not just a membrane targeting peptide. And if you've ever been stung by this ant, you'll sort of be pretty clear on that fact too, that there's something more to it there. But there's also more than just the bullet ant. In Australia, we have an ant called Retita metallica, or the green or greenhead ant. And that's one of the most widely distributed ants in Australia. If you have a grass lawn here in Brisbane, you have greenhead ants in it, and they own it. You don't own your lawn, the ants own your lawn. But anyway, stings from these ants can sometimes cause severe, long-lasting pain. And they cause some other weird things like piloerection, which is just goosebumps, and sweating at the sting site or dampness at the sting site, and a really well-defined axon reflex flare at the sting site, so indicating that your neurons are activated. So the start of this little sub-project, our goal was to take the bullet ant, the greenhead ant, understand what the responsible toxins are behind that pain, and understand how they worked. So the chemistry and pharmacology behind that sting pain. With the bullet ant, we're not the first to sort of recognize this as a unique sting. So we weren't starting from scratch. Others had laid this foundation. So Tom Peck, actually with Justin Smith way back in 91, identified a neurotoxin in the peptide in the venom. And then later, him and another team showed that these were acting on sodium channel currents. And then more recently, Steve Johnson over in the US showed that this particular peptide could modulate a voltage-gated sodium channel. So we had some good clues to start our work with. So what I did was analyze the venome of this ant. And if you look at this, if you're used to looking at HPLC traces or LCMS iron chromatograms, you usually see a lot of different peaks. When you look at this with the bullet ant venom, you basically see one peak. And that peak corresponds to Poneratoxin, which is this peptide here. And you look at the transcriptome and what's found in there, 88% of that transcriptome also corresponds to Poneratoxin. So it's really the major player in that venom, and we didn't have far to look. So we made the peptide, and the assay we do is we look to test whether it can cause pain. We inject it into the paw of a mouse. So this is just a shallow intraplanar injection. We're trying to mimic as closely as possible to a sting as we can. And sure enough, when you inject it, you get immediate near maximal pain behavior that lasts for the course of the recording. And this is completely consistent with what you experience when you're stung by this ant. So we're pretty confident here that the major pain causing component in at least bullet ant venom is Poneratoxin. Greenhead ant was a slightly different story. So we collected the venom, we analyzed the venom as we've been doing, and you can see from this total iron chromatogram there's a lot more peaks than in the bullet ant. And if you analyze that along with the transcriptome, what you see is this. So I don't expect you to read that, just to say that there's over 100 peptides in a pooled venom sample from the colony. And as Ronald actually mentioned before, what we discovered here was that actually this is because lots of individuals with different venom were getting added together. And if we tease out individuals, we see that they actually have different venom profiles, which was kind of cool, but frustrating in some respects when we're trying to identify the pain-causing component. So what you can't see here is that actually those 106 toxins fit into about a dozen different groups. And what we did was we synthesized a representative from each of those groups. So we had about 11, I think it is about 11 toxins there. And we did the same assay, injected them to mouse paw, and we had one huge standout. So what you see here is pain behavior counts over 30 minutes. And one thing that I found really cool here is that this toxin, you see this gradual buildup in the pain behavior. That's also it mirrors what you experience when you're stung by this particular ant. So again, we're pretty confident we've found the pain-causing factor here. And this is this toxin called RM4A. And about this time, Axel, who I'd been working with, Axel Touchard, he emailed me and he said, oh, I've just been on a trip to Cameroon in West Africa. I have this amazing ant called Tetramorium africanum. The sting hurts, in his opinion, more than that of the bullet ant. Why don't we try and figure out what's in the venom? And I said, sure. Yep, send me some of the venom. I should mention that when he told me about the sting symptoms, it all seemed very familiar. It was basically the same thing as Retita ponera or the green ant. So he sent me the venom. I used a different approach. So this is, a third approach to try and solve this problem. So in this case, I had enough venom that I could fractionate it by HPLC. So each of these peaks here represent an individual venom component. You see it's got some complexity to it. And then I tested each of those fractions on F11 cells, which are a sensory neuron cell model. And we had our hits for the active fractions, this F12, F22, and F23. And we synthesized the corresponding toxins, so TA2A and TA3A, one of these. And we found that actually it was one of these toxins, I think F23, which corresponds to TA3A, that caused that pain phenotype in the mouse assay. Oh, sorry. So again, we thought we've come up with the pain-causing factor here. And to give us a bit more confidence, we looked at the sequence of that. And if you align it with RM4A from the green ant, you see that they're clearly similar. And also, if you look at the precursor structure, they're homologous. So again, we're pretty confident we've got the pain-causing toxin from these three ants. So the next question is to understand how they work. So won't dwell on that slide, but basically one assay that's sort of really key to what I do is this one. So as some of you may know, when pain is sensed, it starts when you encounter a stimulus at the end of a sensory nerve ending. The signal travels up this axon or a nerve, and the cell body for that is in the dorsal root ganglia, which sits alongside the spine. And then the signal goes up your spinal cord and you experience the pain in your brain. So these DRGs house your pain sensing neurons. And what we can do is take these out of a mouse and culture them. And so that's what I've been doing. I load them with a calcium sensitive dye, and this allows me to measure excitation basically. And I can look at this. I should mention that not all sensory neurons are the same. There are some that measure, are there for sensing heat, some that are there for sensing other stuff like mechanical stimuli, for example. So it's this heterogeneous population of neurons you can look at down a microscope, and you can image as you add toxins, and I'll show you how that works. So here's a population of sensory neurons. So I can add the toxin. I'll tell you when that's added. So it's just been added now, and you can see that neurons start to light up. And you see that it's actually not all the neurons. So it's a subset of the neurons. So what's happening is this particular toxin here in this example is actually targeting something that's only found on a specific subset of neurons. And you see just there, they all light up. So this is the positive control that's allowing me to see these neurons. And as I said, you can trace all of those cells in real time and see which ones are being activated. So each of these traces here represent an individual neuron. The colored one is the average. And you can also look at the subsets of neurons that are being activated by a given toxin. This can all give you clues about what the target of that toxin might be. You can also add blockers and things or agonists and look at how those can block the response or how the response compares to that. So when we add Poneratoxin, we saw that actually 90% of the neurons light up, which is consistent with the instant pain that you experience. And that if you add Tetrodotoxin, which is a blocker of voltage-gated sodium channels, you can bring that right back down to essentially nothing. So it was consistent with sodium channels. And also we then tested TTX in the mouse model, and we saw that, yeah, it does the same thing. So basically we concluded that our target was TTX sensitive and almost certainly a voltage-gated sodium channel or multiple subtypes of those. So from here, we did some electrophysiology of these. And for those who haven't done EFIS, don't be intimidated by this slide. It's not that complicated, really. So basically what you're looking at here, the black trace is if you imagine this channel is activated by voltage, right? So a depolarizing step change in the voltage. So from -90 to -20. And when it's activated, you get a spike in current, so ions flowing through. And then the channel rapidly closes. So that's what happens there. You get this spike and then it rapidly closes. And that's a normal response to a depolarization for this particular channel, NaV1.7. But in the presence of Poneratoxin, you see that actually that spike is diminished, but the channel is unable to close. It stays open as it's depolarized, and this allows more current to flow through. So the channel is being overactive essentially. And again, here this is a voltage versus current plot, and you can see that usually this channel, the black and white line here, it will activate with a V half of activation around -25 millivolts. So that's how much you need to depolarize to get the channel to open. When you have the toxin present, it opens much earlier, at least a population of the channels too. So you're looking at channels starting to open and current flowing through at -60 millivolts rather than all the way down at -30 or -20. This response does not wash off. So you add the toxin stays on. That's what this plot indicates. And we looked at a range of other NAVs, and what we actually found that sure, NaV1.7, which we know is involved in pain, that's activated by the toxin, but the channel that's most sensitive is actually NAV1.6, and you see that here. You get this huge inhibition of inactivation, huge shift in the voltage dependence of activation, and it does not wash off. So essentially what the toxin does is it creates a channel that's lower threshold for activation, and it doesn't shut. It has a sustained current, and all of this will lead to increased neuronal excitability if those channels are present on a neuron, and they are on pain sensory neurons, and this is consistent with pain. And now when we looked at RM4A, we thought initially we didn't know what to expect, but actually we ended up seeing something pretty similar to Poneratoxin. So again, the channel is unable to inactivate normally. The V half of activation is shifted, so it's much lower threshold, and it doesn't wash off. And again, NAV1.6 is more sensitive to the toxin. And then TA3A, which is the main component from Tetramorium africanum venom. Again, this one did something really a little different. The channel wouldn't inactivate when the toxin was present. And the toxin locked it open. It wouldn't even inactivate once you repolarized the cell. And this was something unique to this particular peptide within these peptides. I will mention in a minute, you do see this with some other sodium channel toxins, some small molecule alkaloids. Again, a massive shift in the voltage dependence of activation, and it does not wash off. And here's just the concentration response curves demonstrating that it's active at 1.6, 1.7, and also a little bit at 1.8. Yeah, so a key question here that I'm really interested in is, and it may not seem that obvious, is did these toxins actually evolve as defensive toxins, or are we just looking at a side product of a toxin, a predatory toxin? Because these ants, they could be using their venom for defense, of course, we know they do that, but they could also be using it for capturing their insect or arthropod prey, paralyzing it and bringing it back to the nest. So I wanted to know, did these evolve under selection pressure from vertebrate predators? And the way I tested that was inject the same toxins, but also some other toxins from these venoms into different insects. I'm showing data here from blowflies that Axel Touchard collected. And what you can see is that, we'll look at the T. africanum one first. So TA3A, which is our pain-causing peptide, did have some activity, but only at really, really high doses. And what it caused there was this slowly developing temporary paralysis. And again, I stress this is at really high doses, and it wasn't lethal. Whereas you take the other component from the venom, TA2A, that we'd synthesized, and that causes rapid, irreversible paralysis that ultimately leads to lethality. PC1A, or Poneratoxin, was basically the same as TA3A, the slowly developing reversible paralysis, but only at high doses. And RM4A from the green ant, our pain-causing toxin, was completely inactive. And this was contrasted by another peptide in the venom, RM1A, which again, immediate irreversible paralysis and death. So what this told us was that on the one hand, clearly in ants, in ant venoms, there are peptides that have different functions. Some of them are there for prey capture, probably, whereas some of them are there for defense. And I believe this data demonstrates that our peptides probably have evolved under defensive selection pressure, almost certainly have, to deter vertebrate predators. And another cool question to ask is, when did these evolve? So I'm not showing all the data we have here, but basically what you can do is, what we did was search for homologous sequences in the venoms of other ants across the ant family tree. And those are indicated in orange where we found them. Of course, some ants no longer sting, so these are indicated in gray. And I should point out, we weren't able to look in all lineages or all subfamilies of ants. Probably should have indicated which ones here actually, but from this, we were able to estimate origins, or the most likely origins for these defensive toxins. And they actually originated way back in the Cretaceous, which I think is kind of cool. And we think they might have originated twice. And this is actually supported by the phylogenetic data from the toxins themselves as well. So Poneratoxin actually groups quite separately to the toxin, the same family of toxins here. So it looks like it's an example of converging on the same function, probably due to similar defensive selection pressures. And again, showed that these clearly evolved at least as early as the mid-Cretaceous. And I think it's kind of cool to think about the reasons why those might have evolved there, that there must have been myrmecophagy or ant-eating, things filling an ant-eating niche back then, and what those might have been. So I'll summarize this here. I've shown that, or others have also observed, that certain ant stings are exceptionally painful. I've shown that peptide toxins are responsible, and that these cause pain via modulation of voltage-gated sodium channels and peripheral sensory neurons. And I've shown that these probably evolved as specific defensive agents. One thing that's, I think, is really cool, these are structurally and functionally unique. They're quite weird peptides, actually. I didn't go into detail, but they're really hydrophobic peptides. They're unique among described neurotoxins and other peptides. And actually, the effects on sodium channels most closely resemble some of these sort of lipophilic alkaloids, such as batrachotoxin from some of these poison dart frogs, which I think is kind of cool. And yeah, to summarize what we've achieved here is we've given new insight into the chemical mechanism of defense by these ants. And what I hope is that these toxins, which have a new mechanism of action on sodium channels, are going to be useful for scientists, including myself and others, to study how sodium channels function, particularly in pain. And last of all, I will thank those involved, in particular Axel. You saw his name come up a fair bit, and the team at the Université de Toulouse, and also the team, my teammates here at UQ who were involved, the late Justin Smith from Southwest Biological Institute, and also Ivan Undheim, who is now at the University of Oslo. And thank you all for listening.
Thank you, Sam. Very interesting. Great picture of a centipede head.
Yeah, it's good, isn't it? Can you see the little droplets of venom?
Of course, I can see the little droplets. You're showing off here. So the floor is open for questions. People, you can, if you're shy, type them in the chat, or if you're not shy, you can just speak up and make a comment or ask a question. I see two people congratulate you on your great talk. Maria has a question, and I will read it out to you. It said, in the first part of your talk, when you injected ant venom into mice, was pain accompanied by an inflammatory response? More generally, do ant venom and toxins cause only pain or also inflammation?
Yeah. So the first question, was it accompanied by an inflammatory response? Yes, these toxins actually do cause quite a strong, maybe not inflammatory, but they cause strong pore swelling. And we're not 100% sure what that's indicative of, but it is dependent on neuronal activation. So it seems to be a neuronal mediated sort of inflammatory looking response. Yeah, and whether or not we see this generally across ants, I think probably almost all crude venoms of ants, if you were to inject them into a mouse paw, you would see some degree of an inflammatory response. But individual toxins, it varies a lot. So I've injected a lot of toxins in this assay, and some cause inflammation, some do not at all.
Cool. Some other questions come in. I will just read another out to you. I think Sebastian de Tertre asks, says, great talk. What are the main predators of ants, and would it be worth testing your peptides on these?
Yeah, that's a good question, and it's something that I've sort of given a lot of thought to. For some of them, say the West African species, I don't know what the predator is, and I don't. It's quite an obscure ant, actually. I'm not sure that that's known. Yeah, for the other ones, I just I'm not 100% sure, and I think there's just not enough basic biology done on some of the stuff, and it's definitely an empty niche there. I think if anyone ever wanted to do it, and it would really complement what we're doing here. But I think another thing to consider is that well, obviously mice are not the natural predator, or at least not lab mice are not the natural predator of these ants. I think they're probably still a good model in that I think there's a chance, and this goes for any venom prey predator interaction, that the natural predator may have developed some form of resistance. And I think if you were to screen or look at that, as has been done in some cases, you'd be getting a slightly skewed picture in that your toxin would look less efficacious compared to, say, a naive target.
Yeah. You would just think as well, I mean, the sort of general pain sensing machinery is pretty conserved, I guess, in most mammals, unless you have these specialists like naked mole rats becoming insensitive to pain associated with acid and stuff because they live in these extreme environments. But yeah, I guess there is ant specialists in Africa as well. I was thinking aardvark, or maybe that's more termite specialist. I don't know. Interesting. Your research opens more questions than it answers, which is the way of good science. If you don't mind, I'm going to read you another question by Juan Carlos Garcia Galindo. He said, "Great talk, Sam. My question is regarding the peptides that you synthesized. Did you consider to do some modifications and address some QSAR studies?" I don't know what that is, but maybe you do.
I'm not sure about QSAR, but SAR, sure. So structure activity relationship. So where you go through.
That's correct. That's correct, Sam. Structure activity relationship studies.
Yeah, cool. Yes, and I've actually done those for one of these peptides, and it's curious. Probably shouldn't give too much away, but yeah. So we kind of know what parts of the peptide are involved in at least efficacy at NaV17, and they're not. How do I put this? They're not specific to a certain part of the peptide. They're actually spread quite well over it, over the peptide. Yeah, so I have all that data. It's just at the moment, it's one of those instances I'm sure people here understand. It's a nice bit of data, but it's tricky to publish by itself. So sort of looking at what to do with that next. But yeah, we've certainly thought about it and have done it.
One more by Andre Michalek. Hi Sam, great talk. Do you have any explanation of why bullet ant venom is so simple in composition, dominated by one toxin, compared to the other two ant species?
No. I think it's I think you come up with all sorts of hypotheses about this, and it's the simplest venom I've ever worked with. I can't think of any that looks like that. It's really, yeah, and I sort of wonder if the Poneratoxin is achieving everything the ant needs. I imagine that the common ancestor of that ant actually did have a more complex venom. In fact, it almost certainly did. And why it's reduced down to a single peptide, I'm not sure. But to me, it sort of seems like a risky strategy, you know. But yeah, I think that's a good question, and it'd be cool to sort of look into for sure.
I was struck. I mean, this is not so much a question, it's my observation, and you answered it. I started Googling immediately when these subfamilies of ants, the one that houses Tetramorium and Rhytidoponera, when they diverged, because when you showed the alignment of that two peptides, that's homologous, right? I mean, the chance that's, and if you say if that is a divergence that could be traced back to the Cretaceous, we're talking about 130 or more million years of independent evolution separating with look almost unchanged peptides. I was just struck how unbelievably conserved that is.
I agree. It's fantastic. On the one hand, it's really fantastic to think that that ancestor of those ants back in the Cretaceous almost certainly had that exact toxin doing exactly the same thing, which I think is really cool, right? But to think that it's retained that in different lineages over that length of time is just mind-boggling, and it does raise a lot of really cool questions. Yeah,
it just shows as well how unbelievably strong the selection pressure on these creatures is to have this pain-causing peptide.
I agree.
It's a defensive gun, because otherwise it would have changed or it would have been lost.
Yeah. No, yeah, and saying that, it has been lost in some lineages of ants, for sure.
True. But
in fact, those two, but not those two. Yeah.
Yeah, but yeah, so it's you know it's phenomenal. I'm gonna, if you don't mind, I'm gonna finish with one more question that was just arrived by Gustavo. He says, "Dear Sam, thank you for your great talk. I understand you're interested in the specific toxins responsible for causing pain, but when it comes to venom evolution, it is the venom phenotype as a whole and not the independent toxins that are under selection. How would you analyze the evolutionary role of the venom phenotype in these species?"
Okay. Yeah, I think I would say that I understand where you're coming from, that it's the whole venom that's under selection, but actually I would say that it's also individual toxins that are under distinct selection pressures in a venom that has multiple functions. I see no reason why different toxins wouldn't have evolved to do different things. So how would you analyze here venom phenotype? Yeah, I'm not really sure. Species. So I think Axel Touchard, he'd be the one to talk to about venom phenotype in these species, and he's done some fantastic work on that. So if you had some questions in that respect, I know that he's done some recent work on this sort of question, if I understand it correctly.
I guess generally we know, of course, that in many different venoms there is synergy in the action between different toxins, and so when you administer individual components and compare their effects with a mixture of them, you can get a different outcome. But it is equally true. I mean, that is known for certain toxins, but it's equally true that there is a subdivision of roles within a venom, especially when you get these quite simple venoms, where certain components are clearly paralytic, it seems, and hence probably prey acquisition toxins, and others are clearly pain-causing in presumably vertebrate predators. There's no real.
It actually brings to mind. It's not just specific to ants either. I can't think of who did the work, but like 50 years ago or more, work on some of the early work on scorpion venoms actually showed this exact phenomenon where certain fractions were paralytic to insects and others were lethal or caused pain in vertebrates. So it does occur across different lineages as well.
Wonderful. So I see that the comments in the chats have ceased. There's a number of other congratulations. So Sam, thanks very much for joining us today at the end of your working day, or what I hope is the end of your working day. It was a great.
It is, yeah.
It was a great talk. Everybody in the audience, thanks for joining us. It's much appreciated. Jan Tietkat raised his hand. So Jan, would you like to say something?
Yes, thank you. First of all, my apologies that I could not join. Due to other commitments in the lab, I was too late. But of course, I'm very interested with this great research. But I do have a question. It has been shown for sea anemone peptides like gigantoxin 1 with a signature sequence having this EGF domain, that they can activate the EGF receptor. It's published, and as such, they couple to TRPV1 activation.
Yeah, I'm familiar with that.
It's published in 2011. So the mechanism already has been proposed in a certain way. And I wonder if you talk about hypersensitivity and hyperalgesia, is not the ultimate, let's say, target, the TRPV1 channel?
Yeah, so I didn't actually talk, I know what you're talking about. This is some other work that I did. on an EGF toxin from an ant venom. I didn't actually talk about that today. I talked about different ones, but I can answer that question. So when I did that particular project, looking at an EGF toxin from an ant venom, we did look at whether it could activate TRPV1 downstream of the EGF receptor in sensory neurons, and we didn't see that for this toxin.
Okay, thanks. That's interesting. Thank you.
All right. Then now, if there indeed are no more hands and no more questions, I think, Sam, we're going to let you off too. Thanks again for joining us. I hope everybody as much as I did. I hope to see you again, all of you, next month. We don't quite know who's going to be the speaker yet, but that will be the last seminar before we break for the summer holiday. So thanks again. Have a good evening, Sam. Have a good day, everybody else. And
right.
okay.
Yeah.